Okay, well, it's a real pleasure to be here and an honor to speak to you. I've tried to take some of the, even the title of my talk might seem a little daunting. I don't know how many of you in the room are neuroscientists. And so I apologize if some of the slides are a little bit dense, but I'm trying to communicate to you just the sort of the state of science in the field right now, and I'm going to focus on some of the things that we've been doing. And I don't think I need to explain to anybody in the room the symptoms of narcolepsy, but just in case, everybody has sleepiness who has narcolepsy, and then the other symptoms like cataplexy and hallucinations and so forth, and it comes on in the teens. And, narcolepsy was first described by doctors. I mean, it's existed presumably for thousands of years, but it was first described by doctors a little more than 100 years ago. And these are the three doctors who really contributed most in the early days. The first descriptions were back around the 1870s, and then around 1916, this guy on the far left, von Economo, he said, I think I know where the problem is, and that it sits in the hypothalamus, a deep part of the brain. He'd seen some patients with injuries there who had really bad sleepiness. And from that time until around the year 2000, that was pretty much all that we knew. It was an incredibly slow time in our understanding of narcolepsy. There was a suspicion that there was something wrong in the hypothalamus, but nobody could figure out how to really crack that question. And then a new neurotransmitter was discovered. It's actually a pair of neurotransmitters. It goes by two names just to make things confusing. Basic scientists refer to this as orexins. Clinicians refer to it as hypocretins. That's probably the term that you guys are most familiar with. And the hypocretin neurons are lost in narcolepsy. In fact, it's really best to think of it as, put it in the plural, because the gene that makes this neurotransmitter actually makes two little signaling molecules, orexin A and orexin B, also known as hypocretin 1 and 2, and they bind to the two orexin receptors. And one of the things that's really striking about this system is that there's only a small number of cells in the brain that actually make it. It's produced in the lateral hypothalamus. And if you look at the picture here, this is a mouse brain. The top picture is the mouse brain cut sort of ear to ear across the front. And those white specks are the orexin or hypocretin producing neurons. And that's basically the only place in the body that this stuff is made. It was only a couple years afterwards that Emmanuel Mignot and Jerry Siegel and others very quickly found that this is the problem in narcolepsy. Those cells somehow die off. This is showing the levels of hypocretin in the spinal fluid on your left, the controls, and that shows that there's a whole range of values in controls, anywhere from the low 200s up to 600 or so. But in people who have narcolepsy with cataplexy, the levels are uniformly quite low. 90% of people who have narcolepsy with cataplexy have very low hypocretin levels. Interestingly, people who have narcolepsy without cataplexy, they can have sleepiness and hallucinations and the whole thing, but they don't have cataplexy. They actually tend to have more normal levels. And we don't really understand that distinction so much. But this really cracked open the field, because now we actually had a chemical in the brain and we knew where it was being produced, and it allows one to then study this in detail. This is showing a similar picture from a human brain. Again, it's sort of cut ear to ear in this direction, and we're just looking at half of the hypothalamus here. This is the base of the brain, right in the middle. It's at the top of the brain stem, but underneath the cortex. And on your left is the normal brain, and at this level you'd see hundreds of cells producing hypocretin. But if you look at the brain of somebody who has narcolepsy with cataplexy, there's only about 5 or 10% the normal number of cells. And so what I want to talk about in the next 40 minutes or so is a couple of the really big questions. Some of these my group has worked on, others have really been contributions by other people. And one of them is the sort of the big thing, which is, the symptom that really drives people with narcolepsy crazy the most, I think, is sleepiness and mental fogginess and so forth. And so we want to try to figure out, how is it that missing this one chemical in the brain produces such persistent sleepiness? The next thing that we were interested in is something that's in some ways sort of almost a really weird curiosity, which is, why is it that for many people who have cataplexy that it's triggered by strong emotions, particularly positive emotions, laughing at a joke, seeing a friend you haven't seen for a long time? What the heck is going on? Why would emotions have anything to do with how your muscles work? Another question is loss of the hypocretin neurons or orexin neurons a sufficient explanation? Is that really all there is to it, or is there something a little bit more complicated? And I think many of you yesterday heard the talk by Jerry Siegel, who was pointing out that in fact there is something going on there beyond just the hypocretin neuron loss. And then if there's time, I want to show you something that has much less to do with science, but much more to do with outreach, which is that over the last year we've been working hard on making a really gigantic website on narcolepsy that will be going public very soon. And so we have some time, I'll show that to you guys. All right, so the first question of why does a loss of this one neurotransmitter make people sleepy? And now, I take care of patients with sleep disorders. I have a lot of patients with narcolepsy, but I spend most of my time working in a basic research lab where we study mice that have narcolepsy. Because now that we know that narcolepsy is caused by a loss of hypocretin or orexin, we can study mice that lack that. And so what we want to do is we want to try to come up with a mouse model that really mimics the symptoms for people with narcolepsy. Now, some of you, in communicating with your doctor, you might have actually seen your sleep studies, and so you might have seen figures that look like this. These are called hypnograms. This is a graph of sleep across 24 hours. And on the left side, you can see the different notations where somebody's either awake, they're in REM sleep, or they're in non-REM sleep, stage 1, 2, 3, or 4. Stage 3 and 4 sleep are very deep unconscious sleep. REM sleep is dreaming sleep. And up top, you can see a typical sleep pattern for a healthy control individual, where they're going to bed around midnight. They have some episodes of REM sleep shown with that magenta color across the night, and then they wake up around 8 in the morning. In contrast, if you just, for somebody with narcolepsy who's just sleeping, as they wish, what you see is a much different pattern. They fall asleep, they wake up, they fall asleep. The nighttime sleep is fragmented, and there's lots of naps during the daytime. So that period of consolidated wakefulness that's occurring in the top individual is really disrupted. And not only that, but the REM sleep is occurring at all times of day. In people who don't have narcolepsy, REM sleep or dreaming sleep pretty much only occurs during the night. It almost never occurs after 8 o'clock in the morning unless somebody's a shift worker and their sleep schedule's all messed up. But in people with narcolepsy, the brakes that normally hold REM sleep in check are broken. And REM sleep can pop up at all different times of day. And that actually is one of the things we think that gives rise to the hallucinations and the sleep paralysis and the cataplexy. And so there's two major problems that we can see here. One is difficulty maintaining consolidated wakefulness across the day and poor regulation of REM sleep. Now, the pathways in the brain that promote wakefulness can be broken down into two broad categories, and I'm just going to go over them really quickly here. In green are neurotransmitter systems in the brain that make what are called monoamine neurotransmitters. And these include things like norepinephrine and serotonin and dopamine and histamine. And all of these project up throughout the whole brain, and they help put the cortex and other parts of the brain into an active waking state. Acetylcholine is also important, and that's shown in blue. And so the cells that make it are shown with the circles, and the arrows are showing the different parts of the brain that it goes to. And the specific nuclei and so forth aren't terribly important right here, but suffice it to say there's a whole bunch of redundant systems that promote wakefulness. Now, when you go to sleep, there's a system that actually puts you to sleep by coordinating the inhibition of all those different wake-promoting systems. And we don't understand this fully, but one area that we think is pretty important sits up at the front end of the hypothalamus. Now, I don't have a pointer here, but the hypothalamus, if you see where that red circle is, that's the front of the hypothalamus. And the hypothalamus extends back towards kind of where the arrow takes a curve down to the brainstem. It's that little base part of the brain. And at the very front end of the hypothalamus is a cell group that was described about 15 years ago. It goes by a long name of ventrolateral preoptic area. And these cells are really unusual in the brain. They're one of the few cell groups that's actually more active during sleep than during wakefulness. And unlike the other signaling molecules we just talked about, which are generally excitatory, these make an inhibitory neurotransmitter that turns off other neurons, and it's called GABA. And these cells release GABA, and they turn off the wake-promoting systems in the brain when you go to sleep. And so you can look at this in terms of a very simple circuit. On the right side of the screen, you have the wake-promoting systems. We call these the monoamine neurotransmitters or acetylcholine. Those promote wakefulness. On the other side, you've got the ventrolateral preoptic area and other cell groups that produce sleep. And what's interesting is these two cell groups inhibit each other. So when you're awake, the wake-promoting systems turn off the sleep cells. And during sleep, the sleep cells turn off the wake cells. And you end up with a situation where normally you should be fully awake or fully asleep. The problem with this system is it doesn't necessarily keep you awake, and nor does it keep you asleep in its own right. And what we think is going on is that the orexin or hypocretin neurons play a really important role in helping tilt the balance here. And they do that by exciting those wake-promoting cells. So we've now put orexin at the top of this diagram. And one of the things that's unusual with the orexin or hypocretin neurons is that they are auto-excitatory. And so when they start firing, they actually get themselves firing even more. And so they have a sustained pattern of activity. And we think that that sustained activity then drives sustained activity in the other wake systems. This is as hard as it gets, by the way. We don't have no more circuits after this. But this, I think, is the essence of it, because if you really think of the orexin neurons as helping produce sustained wakefulness, then you can appreciate that if they're gone, you're not going to be able to stay awake for long periods of time. So one of the questions that we have been working on is trying to figure out, well, what are the key spots in the brain that orexin or hypocretin acts through? Because if we could figure those out, maybe we could develop better drugs that actually target that site better. It's a little tricky because the orexin hypocretin neurons project to all of those areas that promote wakefulness. The monoamines in green, and in purple here, the acetylcholine cells. So to give you an idea of how we might study this, we can do long sleep recordings in narcoleptic mice. And in this case, what we did is we made a new line of mice that are born lacking one of the really important orexin receptors. It's called the OX2 receptor. And so these mice are born with none of this receptor, and they're sleepy. And so let me just sort of talk you through this graph here so you understand how this works. So mice are nocturnal, so they're normally running around a lot at night, and they sleep mostly during the day. And they have long periods of wakefulness, just like people do, but it happens at night. And so if you look at the amount of wakefulness and break it down into either short awakenings, medium awakenings, or long awakenings, what you can see here is this is a graph showing the length of a wake bout as it contributes to the amount of wakefulness that happens at night. And so wild-type mice, the WT here, those are normal, healthy mice. About 60% of their wakefulness is occurring in very long bouts. But the mice that lack one of these orexin receptors, they can't do that. They have the same total amount of wakefulness, but it's occurring in shorter bouts. They can't produce that sustained wakefulness. Now, one of the things we did with these mice is we made them in a way where we can actually pop the receptors back into specific parts of the brain and ask, where do we need to put the receptors to actually rescue these guys? This is a bit of a not so pretty picture, but it gets the idea across. At the top left is showing the normal pattern of the orexin receptor. And you can see there's kind of a hazy pattern up in the top of the brain, the cortex, and a very dense swath at the bottom. Do you guys see that little C-shaped crescent at the base of the mouse brain that's labeled normal there? That's where the histamine neurons are found. And so on the top left is showing the normal pattern. On the top right is showing the pattern when the receptors are gone, and that's just sort of background staining. And on the bottom right is showing us rescuing the receptor expression just in that part of the brain where the histamine neurons are. And so you can see that crescent of orexin binding has come back again. And the consequence of that is that when we put the receptors into just that part of the brain, the mice can now produce long bouts of wakefulness again. So that's really a nice example of how orexin signaling through those histamine neurons, or something in that region, is sufficient to actually help these guys wake up. And so this actually provides a lot of justification for why, some of you might have heard talk about a new kind of wake-promoting medicine that's under development that enhances histamine signaling in the brain. There's nothing like that clinically available yet. And this provides really good argument for why we want to ramp up that histamine signaling, because it's pretty clear that that's a great way of producing long bouts of wakefulness, at least in mice. Okay, so it gets easier after this. I think many of you have seen cataplexy before. You've seen it in yourself or family members. This is a kid who has just horrible cataplexy. When he falls, he goes down hard. And like many people, it comes on with strong positive emotions. He's Czechoslovakian, which explains that Slavic dance there. There's a little editing glitch coming up here. There's going to be a gap in the film. But you'll see, this is cataplexy, right? This is somebody gradually losing their muscle tone, and they can't hold themselves up and slumping to the floor. Now, in his case, like with many people with narcolepsy, it's triggered by strong emotions. And we wanted to study this in mice. Now, when we first started studying these narcoleptic mice, we called these behavioral arrests because we weren't really confident that they were cataplexy, but we're pretty confident now. And so this is showing a cage of 5 mice with narcolepsy. These mice all lack orexin hypocretins. There's a couple mice sleeping in the top left. Watch this guy with the pink arrow. Did you guys, I don't know if you've noticed that, but he was grooming and then he just suddenly stopped moving. This is the same paralysis as happens in people. Of course, when a mouse falls, it's not quite so dramatic as in a human. And if you watch, there's the two mice at the bottom of the cage right now, and they're kind of interacting, they're sniffing each other, they're running around. And, many of you probably appreciate cataplexy comes on under a lot of mostly social conditions. It doesn't really happen much when you're alone. These two guys now have cataplexy at the same time. And it's possible that there's actually some little sort of sympathetic thing going on here socially. And here's this mouse on the left is starting to recover. He's wiggling his hind legs a little bit. And just like in people, when the cataplexy runs its course, which takes about a minute or so in the mice, their power comes back. And this, you'll see here, he actually gets right up on his hind legs again. And so like in people, it comes on pretty quickly, and then it abates pretty quickly. The thing that's a real challenge for us is this. So, I think many of you have probably seen the videos of the narcoleptic dogs at Stanford. And when a dog is happy, it's really easy to appreciate it, right? They're wagging their tail. You can just tell by the way they're moving that they're really having a good time. But mice don't do that. There's actually some rumors, there may be some ultrasonic vocalizations that are higher than humans can hear, where they communicate with each other, but it hasn't been sorted out yet. And so we have to take our best guesses about what would make a mouse happy. And so we've played around with a few things here, and we've actually, over the last couple of years, we published a few papers where we found a few things that actually can increase cataplexy, and thereby we sort of would guess that these are things that may make them happy. One of them is Fruit Loops. Peanuts do it too. Both of these can double the amount of cataplexy that happens. Running wheels. Anybody who's had a pet rodent, you know how they love to run at night? And rodents love running wheels. They will work hard to get access to a running wheel. It's fun for them. And interestingly, putting the mice together, that group housing, the social interaction, presumably there's something positive about that comes out of it. And so these things produced about a 2 to 3-fold increase in cataplexy. Unfortunately, cataplexy is still reasonably uncommon. We'd only see, 6 or 10 episodes per night, which isn't really enough for us to study, and we needed something that was a stronger stimulus. And so this is what we discovered. So we found that if you put one Hershey's Kiss into the cage of these narcoleptic mice at the beginning of the dark period, their active period, it can increase their cataplexy 8-fold. It's huge. And so the graph on the left side there, the white open symbols are showing the amount of time that's spent in cataplexy when they just have their normal chow. The green symbols are showing what happens when they have access to chocolate. And it's just incredible how much cataplexy they have. And what's interesting is you notice the bar at the bottom, it goes from 7 p.m. till 7 a.m. That's the dark bar. And that's when it happens. And then in the light period, they're back to sleeping again. And so it's something that happens during the nighttime, which is their main wake period. So now, some of you might have heard, as you read about the brain, about the amygdala. And most of the literature on the amygdala is focused on fear and anxiety. But the amygdala actually is a part of the brain that also regulates positive emotions. When somebody is really happy, doctors can do functional imaging and actually see that the amygdala can get turned on in some circumstances. And the thing that makes that particularly interesting is that the amygdala is connected to parts of the brain that regulate emotions. And it also is a really important relay for sending those signals down to the brainstem, to parts of the brainstem that actually regulate muscle tone and power. And so it's in a nice location to connect emotions to muscle power. Jerry Siegel, who you heard yesterday, recorded from amygdala neurons in a paper about 10 years ago. And what he found is that if you study them in narcoleptic dogs and stick an electrode in there, you can actually see that some of those neurons are active. So the very bottom trace is showing the amount of muscle activity. That's the squiggly black line. And that place where it goes really low and flat, that's cataplexy in the dogs. No muscle power. And the top line is showing the firing rate, that the electrical signals in some of those amygdala neurons. And so they go up while the cataplexy happens, they come back down again when the cataplexy stops. And so that made us think that the amygdala is going to be a pretty important spot. And so what we found was that if we take these mice and make a little injury that's limited just to the amygdala, that it reduces cataplexy by about 50%. It does it under boring baseline home cage conditions. It does it when we give them running wheels, and it does it when we throw in some chocolate too. And so this makes us think that the amygdala is really probably a necessary part of the mechanisms. It's not the sole thing, because the cataplexy is still happening, but it seems to be a pretty important regulator that's necessary. Now, I have to say we were actually a little disappointed with this. We were hoping that we were going to see a normal amount of cataplexy under the baseline conditions where there's not a lot of emotional stimuli, and yet the cataplexy was reduced there. So we think that the amygdala is necessary as part of the, if you will, mechanism for producing cataplexy, but we needed to look a little harder to find something that was really related to the emotional part of it. But here's how we tuck the amygdala in, and I'm sorry, this is another circuit diagram, but on the top these are drawings of mouse brain. Looks a little different than human brain, but on the left is the front. They have this gigantic olfactory bulb because they're very olfactory animals. They get a lot from smells. On the right side of the screen is the brainstem with the cerebellum sitting above. And there's a place in the brainstem called the sublateral dorsal nucleus, SLD, which you can consider as the master switch that turns off muscle power. When you're in REM sleep, the sublateral dorsal nuclei neurons turn on and they make you paralyzed. That's normal, but we think those cells also turn on in cataplexy. Normally those cells are held in check by this place. The name is just atrocious. It's the ventrolateral periaqueductal gray / lateral pontine tegmentum. It's not even worth saying twice. But those cells do the opposite. They are the brakes on the SLD neurons, and so they're the thing that keeps the muscle paralysis from happening. If you get an injury of those ventrolateral periaqueductal neurons, muscle paralysis starts popping up at all kinds of bad times. Now, the orexin neurons normally excite those cells, the VLPAG cells that help stop paralysis, but the amygdala neurons do just the opposite. And so you have this little push-pull system where the amygdala is in a position to turn off the brakes, but it's being countered by the orexin neurons. The trouble is, if you've got narcolepsy and the orexin neurons die off, that system's not in balance anymore. And so now emotions are no longer counterbalanced by this, and it can turn on this paralysis pathway. But as I said, this wasn't a completely adequate explanation. And so we then said, well, what's happening up in the cortex? And we went to a part of the cortex that's long been felt to play an important role in emotional regulation, and this is called the medial prefrontal cortex. And so the little photo you can see in the top right is another section cut this way through a mouse brain. And that brown area on both sides is where the medial prefrontal cortex is. And what we did is we put a special channel in there, something that can regulate the activity of neurons. And the cool thing about this channel is that it's only activated when we give the animals a drug called ivermectin. It's abbreviated here IVM. So if we put that channel in, and we do nothing else, the animals are completely normal. If we give them ivermectin, those cells in the medial prefrontal cortex get shut down. In fact, they actually get shut down for about 3 days. It's a very long-lasting drug. So let me just walk you through what we did in this experiment. We recorded the baseline activity of these animals with and without chocolate. We then inactivated that part of the cortex, did the same thing again. Then we let them recover a couple of weeks and did the same thing again. And what you can see here is the number of cataplexy. Let's just focus on the left side. The number of cataplexy bouts at baseline is very similar, whether they're in the regular first vehicle or baseline condition, whether they get ivermectin or vehicle again. But then, if you compare that to how they go with chocolate, what we think is the emotional stimulus, vehicle, very high levels of cataplexy, ivermectin, blocking those cells in the cortex, knocks it down, and then when we let them recover, and now they're just getting the vehicle plus chocolate again, they're back up to that same level. And so what this is now showing is that that part of the brain is again necessary for the production of cataplexy. The stuff on the right is just sort of control things, saying that inactivating that part of the brain didn't affect wakefulness and didn't affect how much chocolate they ate. And so now this is another cell group that we think is important for regulating cataplexy. We can add it to our little circuit diagram. So here's the same things, the orexin, hypocretin neurons, the amygdala, and now the medial prefrontal cortex sits up above it. And so what we think is going on, it can be summarized in the bottom panel there, where the medial prefrontal cortex is the part of the cortex that's probably responding to these social cues. It probably helps distinguish whether something's actually humorous or whether it's really annoying, but it's involved with producing these strong emotions. It then turns on the amygdala, and the amygdala then activates those brainstem pathways that eventually result in the cataplexy. This needs to be confirmed in humans. We've only studied this in mice thus far, but we think it's a reasonably good first explanation for how emotions might trigger cataplexy. Okay, so now I think this is a story that you might have heard about yesterday, which is narcolepsy caused by more than just loss of the hypocretin neurons? We talked before about how the hypocretin neurons project to all different brain regions, and honestly, for the longest time, we just said, okay, fine, this is a sufficient explanation. We know that losing the hypocretin neurons in mice is enough to basically make them sleepy, make them have cataplexy, so end of story. Maybe that's all there is to it. And we were working on a project actually to a completely different thing. We were looking at the hypothalami of people who have traumatic brain injury. And the postdoc, Philip Valco, who had been working on this, had finally, after 3 months of work, found a way to identify the histamine cells in human brains. It was actually technically really hard. And he said, let's look at some narcolepsy brains. And the reason was because there's been some talk about altered histamine signaling in people with narcolepsy. This is a paper from Kanbayashi and Nishino a few years ago, where they measured histamine levels in spinal fluid, same place that we measure hypocretin levels. Now, I'll tell you, it's actually really hard to measure histamine. It's a very labile, unstable chemical in spinal fluid, and so the levels, even in normal people, are all over the place. So you can see on the left, neurologic controls. The levels run the whole gamut, but there's some low, medium, and high. But if you look at spinal fluid from people who have narcolepsy with cataplexy, the levels are generally low. Same thing for narcolepsy without cataplexy, and same thing for idiopathic hypersomnia, which is a similar disease causing sleepiness. But if you look at patients with sleep apnea, the levels tend to look more like the normal range. Now, this is kind of controversial because there's been a couple more papers since then that have said maybe with a more sensitive assay this isn't true or not, and honestly it's sort of undecided yet whether histamine levels are low or normal, but it was still an interesting clue that the histamine system might be messed up in narcolepsy. And so, as I said, after a lot of work, we got this procedure working where we could actually look at this in the brains of people with narcolepsy who have died or in control brains of people who don't have narcolepsy. And this shows the pattern of where the histamine neurons are. We're staining for something called histidine decarboxylase. It's the enzyme that makes histamine. And the bottom circle, which is kind of pale on the left-hand side there, is called the mammillary body. And the histamine neurons are smeared up and around this. And in narcolepsy, we see the cells in the same place. But after Philip stained a couple of these brains, he came to me and he said, I think there's more cells. I said, that can't be right. That makes no sense. Why should there be more cells? I said, go back and try a couple more. And he did. And it came back the same way. And eventually he counted cells in 7 brains from people with narcolepsy and a dozen control brains. And the overall pattern that we found is, not surprisingly, the orexin neurons are lost to a great degree. There's a 90% reduction in the number of orexin hypocretin neurons, but there was a 94% increase in the number of histamine neurons. This just came out of the blue. We were really, really shocked at this. And there's another group of cells that are found in the same area that make something called melanin concentrating hormone, or MCH, and those are unaffected. They don't have much to do with the control of sleep and wakefulness. But we were really surprised to find this. In fact, we were so surprised that I had another postdoc independently go back and completely repeat all the cell counts just to make sure there wasn't any bias in it, and he came up with the exact same result. And of course, you just heard yesterday that Jerry Siegel group has found something very similar. In the brains that they studied, the increase wasn't quite as large as this, but it was still huge. I think he found about a 60% increase, something like that. So just like their group did, we went on to check this in narcoleptic mice to see if the same thing is going on. And we found something a little different. They did not see any differences in the mice, and we did. And so, again, the orexin, we looked at 3 different kinds of mice. One is healthy, wild-type mice. In the medium blue are mice where the orexin hypocretin neurons die off over time. And then in the purple are mice that are born with no orexin hypocretin at all. And we found, of course, the big loss of orexin neurons on the left. The histamine neurons were increased in the mice, especially in the mice that had never had hypocretin, and the MCH neurons were unchanged. So this is where the Siegel group and we diverge. They said that the mice that they studied seemed to not have a change in histamine cells, and yet we see this change. So I can't explain that just now. But the important thing is for people, we both find a very, very similar phenomena. And so then we're left with these questions of what could cause an increase in the number of these histamine-producing neurons. Well, it turns out that the histamine neurons get this incredibly strong excitatory input from the orexin hypocretin neurons. And it's possible that when those cells die off, the histamine neurons are saying, what's going on here? And they're trying to compensate in some fashion. They're normally accustomed to getting a really strong excitatory drive, and yet it's missing. And so they may be trying to bounce back, and they may increase the production of the enzyme that makes histamine, this histidine decarboxylase. It's also possible that there's cells that normally don't make histamine at all that sort of switch and become histamine-producing cells. And then I think this is the least likely, but it's still an interesting idea, is there maybe even birth of new cells that are part of this compensatory process. I think the mechanism is interesting to me. The consequences are more interesting to you guys. And what does this do in the brain, and does it actually have some adaptive or perhaps maladaptive significance? And as I said, normally the orexin neurons strongly activate these histamine cells. And you could imagine that if the orexin neurons suddenly die off over the course of weeks or months, histamine signaling may be reduced because they're not getting driven and activated in the same fashion. And so it may be that as the brain recovers, this increase in histamine may do a couple of things. Now, I don't know how many of you either had narcolepsy starting at a young age or have a kid who started at a young age, but the story that I hear oftentimes is that particularly in like pre-pubertal kids, when they get narcolepsy, sometimes they sleep a ton. They go from sleeping 9 or 10 hours a night up to 12 hours a night. And in adults, we don't see that excess sleep. We sometimes call that hypersomnia. And so it's possible that if the histamine system is underactive for some period of time at the onset of narcolepsy, that's actually contributing to these really large increases in sleep. But as that system compensates, it may bring you back to the normal, 8 hours of sleep or so. It's also possible that this change in this system is one of the factors that causes the weird conditions that we see in narcolepsy, where people have the paralysis of REM sleep, like sleep paralysis or cataplexy, yet they're still conscious, because we know that the histamine neurons help keep you conscious. And so if that system is now ramped up somehow, it may keep you conscious even when you're in a REM-like state. So this is just speculation right now, but these are possibilities. There's so many questions. There's lots of things we'd really love to pursue. And before I forget, I would just sort of put in an appeal. I think you've heard this from Dr. Mignot and Dr. Siegel, and now you're going to hear from me. We need more brains from people with narcolepsy. I know it's not really great lunchtime conversation, but particularly people who have well-documented narcolepsy without cataplexy. We have a nice system in place in New England for collecting brains. We just got one last week, actually, from an 82-year-old person who died of other causes, and she had textbook narcolepsy, and we're going to learn a lot from that. And so, it's great to speak to your family members, if this is something you wish to do, and you should. And it's probably, if you live in California, it's probably easiest to contact Mignot. If you're in New England or the Northeast, talk to me about it, because we would love to get people. We have like 20 people signed up right now, we're doing pretty good on controls. But if you want to sign up as a control, that's okay. So one of the huge question is, what actually kills off the hypocretin neurons? We think the immune system goes haywire, and I think Dr. Mignot is going to have a pretty big story on this soon. But we're increasingly confident that something goes wrong with the immune system where it goes bonkers and it kills off the orexin hypocretin neurons. But when it comes to narcolepsy without cataplexy, sleepiness, hallucinations, sleep paralysis, the whole kit, but not cataplexy, we know so little about that. The world's literature consists of 1.5 brains. That's all and that was from Jerry Siegel's group. And so we really, really need to learn more about that. We don't have a clue as to what causes sleep paralysis or the hallucinations. Those are big mysteries. And what about the symptoms that really bother people? Like, a lot of folks with narcolepsy, yeah, they're sleepy, but they're also just struggling to get through the day because they feel in a fog, they can't pay attention for long periods of time. And what about those cognitive aspects? That's something that really needs more work. And other questions like, why is it, particularly in children when they get narcolepsy, that they sometimes put on weight? This whole issue of obesity is really a very big mystery, and there's lots more questions. And so, just to wrap things up on the science part of this, I just want to emphasize the main points. We talked about how the orexin hypocretin system, we think that one of the really important spots that it acts in the brain is those histamine cells. I showed you some evidence that at least in mice, the medial prefrontal cortex and the amygdala seem to be necessary for cataplexy, because when we mess those up, the cataplexy is reduced. And then this last finding, the most sort of mysterious one that we haven't really sorted out yet at all, is this increase in the histamine neurons. How does that happen? What's the consequences of it? So I run a lab. I don't actually do a lot of the experiments. I try to point people in the right directions. The folks who really did the work, Philip Valco did that human brain work. Christian Burgess, Yo Oishi, and Takatoshi Mochizuki did a lot of the mouse work that I just showed you. Now, before we finish up, I want to switch to something completely different. I think we have a few minutes. Yep, we got 15 minutes. This is perfect. So there's a little time for questions, but I want to just tell you guys about something which is a new project. For the last year, we've been at Harvard Medical School, we've been making a website that's all about narcolepsy, and it's really oriented towards lay people. It's got some science in it, but it's mostly about all different aspects of narcolepsy. If you actually read through the whole thing and watched all the videos, it would probably take about an hour and a half. And so this is going live in November. And I have a whole bunch of cards with the web address that we can give out if people are interested. And it covers things like, first explaining what's narcolepsy, what's it like to live with narcolepsy, how is it diagnosed, how is it managed, and we've got some really nice videos that go with it. Anyway, the videos are great. Nicole Giuray's video is really, really moving. It's talking about her life with narcolepsy and how it impacts a lot of different aspects, but that's part of who she is. Let's go straight to the questions now. Does anybody want to talk about the science of narcolepsy, or there's lots of questions on medications this morning. We didn't get to half of them. Yeah? So the question, if I understood it, is there any variation in histamine levels between people who have plain old narcolepsy and narcolepsy where there may be some immunologic issues like allergies and so forth? I haven't seen anything that really distinguishes between those. And as I showed you, I think the whole issue of measuring histamine is really in its infancy right now. There's a tremendous amount of controversy about how to do it and how to get precise and really reproducible results. There's 5 papers that I'm aware of where people have measured histamine in relationship to sleep disorders. 2 of them say levels are low, 3 of them say levels are normal. It's kind of a mess right now. I think we need to wait until we actually get better at it. The autonomic nervous system is the part of the brain and spinal cord and so forth that's involved in these sort of unconscious things like sweating. It also makes your heart go fast, makes you breathe fast. And the autonomic nervous system gets activated by any sort of stimulus that has a lot of intense emotions. And so a lot of people with narcolepsy have very intense dreams. And I think what you're describing is that the dreams are so intense that they're actually causing that activation. So just like if during the day something scared you tremendously, you'd start sweating, the dreams are probably of that same emotional intensity that it can cause the sweats. I guess I would also just mention that, there are some other things that can get worse in REM sleep in association with dreams, like sleep apnea. Sleep apnea can get worse, and sleep apnea can make people sweat too, just from that. So it might not just be the dreams itself.
No, I have a machine.
Uh-huh. Yeah.
I've had the sweats before I was diagnosed, but now.
Yeah, well, if it's set up properly, then it should cover you through REM sleep as well. But it might be, you should just talk to your sleep doc about that. Yeah. The question is there any connection between narcolepsy and autonomic dysfunction? And even though narcolepsy is we think it's an autoimmune disease, there's never been any proof that other autoimmune diseases show up with any higher prevalence. And so, which is kind of a weird thing because for some autoimmune diseases, they kind of come in clusters. Like somebody may have thyroid dysfunction plus arthritis, and they show up with a greater prevalence than you'd expect. But there's really no evidence for that in narcolepsy. So in this particular, and I haven't actually seen much literature suggesting autonomic dysfunction with narcolepsy. I would say it's probably just bad luck. Yeah. Other side of the table there? Yeah. So I think if you go to the textbooks and look at the scientific papers, you won't find much mention of narcolepsy changing over time. The idea is that in the few months after it begins, what you've got is what you're going to have. However, there was a paper that came out from Mignot's group last year. The first author on it is Andlauer, where they noticed that, I don't know if you remember, I showed a scatter plot earlier that showed that amongst people who have narcolepsy without cataplexy, there's a small number, about 10 or 15%, who actually have low hypocretin levels. And if you follow those folks over time, about a third of them will actually eventually develop cataplexy. And that's probably pretty good evidence for some progression of the disease. And so the message that I tell folks is that in general, one should not anticipate much worsening, but I don't think it's impossible. And I think that it is possible in some people for it to evolve over time. The one good thing that I'd mention in association with that is that if we can really figure out what's actually hurting the hypocretin neurons and damaging them, then that actually means that there's actually some hope to get in there and stop that process if it really is extending for months or years. Yeah, you had a question with Gray?
Yeah,
Right. So the question is, a lot of the diagnosis of narcolepsy now relies upon sleep tests. And sometimes, particularly the multiple sleep latency test, or MSLT, the daytime nap test. And in some patients, we see a very striking pattern. We're like, oh yeah, that's definitely, it fits with your history, it's definitely narcolepsy. But ask David Rye right here at the front table. He'll tell you how these tests are not all that reproducible. You do it one time and then you do it a year later and it looks different. And the trouble is, and I think this is getting to your point, we're not really measuring what's going on in the brain. We're measuring on a macroscopic level what somebody's behavior is like, and there's so many things that can influence it. Like, what if when it's your first night in the sleep out and you had a rotten night's sleep? Of course you're going to be really sleepy the next day. That may not be really reflective of you in general. And so what we would love to do is have a biomarker, some blood test, some spinal fluid test, where we can say, that's definitely narcolepsy or it's definitely not. That's something we don't have right now. I should really let him sort of take credit for saying this, but you may be hearing in the next year from Dr. Mignot about how he thinks that he may be finding some immune cells that are uniquely activated in patients with narcolepsy. And if it really is specific to narcolepsy, that would be great, because if that really is the case, that would be a blood test. Probably an expensive blood test, but still. But it would be. We would love to have that definite handle where you're really getting in close. The ideal thing would be, as you say, to measure hypocretin. But there's not a lot of people who are really gung-ho about having a spinal tap. It's not the worst thing in the world, but it's a barrier. Yeah. So I think, is your question about dementia? Yeah. So I don't know of any evidence that having or not having narcolepsy increases your likelihood of getting dementia. I think there's a lot of different angles to this. There was a paper by David Holtzman a couple of years ago where you could read it in such a way as to think that having low hypocretin might actually be protective because your brain is sort of not running at full speed all the time. You may not be sort of tiring out your neurons in the same way, and that may be somewhat protective against dementia. But Jerry Siegel and our group teamed up, and we basically came to the sort of, it was not a great study, but our feeling is that dementia is just as common in people with narcolepsy as it is in the general population, at least Alzheimer's disease. It's not been looked at in any sort of thorough way. But I think the question also ties into this thing that's just where we don't have great answers, which is, does taking stimulants for 40 years have any consequences for the brain? And we don't really have good answers on that. If it's harmful, it's not harmful in a really huge way, but it hasn't been studied as much as it could be. Anyway, I'll just tell you, it's a great reason to go to the website. It should be coming out in mid-November. And I will put a bunch of the cards out at the front here, so as you guys leave, you can take one of these. It has the web address on it. I would really encourage you to look at it. And I have to say, there's a few things on the website that there's not much in the literature. We have some nice little sections on issues around narcolepsy in teen years, in the working years, in elderly people, in pregnancy, issues around narcolepsy and driving and so forth. Julie Flygar helped write some of the content for it, and I think it's got a nice balance between science and real life. Yeah. So the question is, does the changes in the hypocretin neurons or the histamine neurons, how quickly do these things happen? And the challenge for us is that we only really can study this system, at least under a microscope, when somebody dies at the age of 75 of some other cause. And so we know extremely little about what happens right around the onset of narcolepsy. And the challenge is made even worse by the fact that for many people, it takes months, if not years, to actually get the diagnosis made. And so what we really would love to do would be, in the case of like spinal fluid, maybe if we do spinal taps within a few weeks of the onset, if that were possible, maybe we would learn something at that point. But there's very, very little data about the immediate period around the onset of narcolepsy. So it's a huge gap in our knowledge. So if I heard you right, is there any way of increasing the number of operating hypocretin cells? Is that right? So yes and no. We have a way. We did it in mice. We have a paper in the journal Sleep that just came out a couple months ago, where we took it's called a viral vector. It's basically, viruses have a protein shell and then they have all their DNA on the inside. We took all the virus DNA out and we put the hypocretin gene in, and we squirted this into the brains of narcoleptic mice, and they did great. They went from being very sleepy to having almost no sleepiness. But it requires squirting a virus into the brain. And so, but that it's called virally mediated gene therapy, and this technique is being used for really horrible fatal diseases, the kind of diseases that kill kids, things like Parkinson's disease, where somebody may have only a couple years to live. And so people are starting to use these kind of gene therapy techniques to improve neurologic diseases. personally, even though we have a technique that works in mice, I think what I'm going to do is wait a few years before we actually decide whether to push this on into dogs or monkeys and maybe even into people. Because right now I think it would be hard to justify the potential risks of putting a virus in the brain versus the benefits. There's a few patients with narcolepsy where even despite optimal medications, they're really having a hard time, and those might be the first ones that we treat this way. But I would say that's years in the future.
I think
Should we wrap it up?
continue.
Well, thank you all for the good questions.